Hello, my name is Zachary Flamholz, and I am a member of the Kelly Lab in the Systems and Computational Biology Department at the Albert Einstein College of Medicine in New York. And I'm here today to talk about our project using large language models to improve annotation of viral proteins. As a quick overview, my talk will consist of the background and the motivation for our project, the design of our experiment, and some of the results that we've had so far. Over the last number of years, with the increase in metagenomic sequencing, there's been a desire to catalog the viruses in different environmental ecosystems or across ecosystems based on aggregating data from multiple studies. And here we're looking at 5 data sets, 2 from the human gut, 1 from the ocean, and 2 catalog studies from across systems where the viral genomes were aggregated, their proteins predicted, and protein families were constructed by aligning the proteins in those genomes. And what we're highlighting in this figure is the fraction of viral protein families that received any annotation through automated annotation mechanisms. And this annotation is primarily done through sequence homology. To address the viral protein annotation gap, we leverage developments in the field of natural language processing, which are increasingly applied to biologic sequence data. Briefly, a language model is the probability distribution over any sequence. These models are trained in a self-supervised manner, meaning that they are trained on data without labels, and this allows them to leverage vast amounts of training data. And intuitively, we can think of an analogy for natural language where an amino acid is a word and a protein is a sentence. Language models trained on protein sequence data have been termed protein language models, and they've been shown to capture physicochemical properties of amino acids and even sequence of amino acids in the forms of structural domains and taxonomic relations at the level of whole proteins. So in thinking about how this technology can be used to do remote homology detection for viral sequences, we can contrast our approach against the existing method of sequence homology, where sequences are aligned one against each other. Alignments are used to form multiple sequence alignments that are converted to statistical probabilities of amino acid sequences, which are in turn converted to hidden Markov model profiles. And a sequence is considered to have matched a profile if it statistically aligns with the hidden Markov model profile. Our approach to leverage language models for functional annotation allows for the conversion of a protein sequence into a learned representation or an embedding, and then we can train a classification task over functions to predict the protein function. For our experiments, we leverage the frogs database that has 2 properties that make it suitable for our experiments. The first is that it was designed for remote homology, and the way that this was done was hidden Markov model profiles of viral protein families were iteratively collapsed if profiles matched other profiles statistically, and then they were then rerun to see if profiles could be collapsed further. Additionally, the frogs database team manually curated 9 protein family categories that were assigned manually to viral protein families after automated annotation. And these high-level functional categories were what we attempted to determine whether a language model classifier could predict. So the core structure of our experiment was whether or not the functional annotation of a family could be predicted after training on other families of that functional annotation. So we took all of the families that had annotations and we trained a classifier where in each fold of training, 80% of the families were used in training and 20% were held out in test. And for the test families, 1 single sequence was evaluated for its categoric functional assignment. In training, all sequences from a family were allowed to be trained on, and in testing, a single sequence. And what you'll see from the performance, though different across categories, the classifier was able to identify with high predictive capacity the correct categoric assignment. As a follow-up experiment, we sought to determine whether protein language model pre-training, dataset, model architecture, and training objective would affect the performance on the viral protein function classification task. We evaluated 4 models, 2 from the PROS project, 1 from the Protein BERT project, and 1 from the PROT-Trans project that had varying architectures, sources of proteins, and training objectives to determine the performance on our classification task. And we observed that the PROT-Trans model, which was crucially trained only with the mask token objective and on the much larger BFD protein database of over 2 million protein sequences, performed overall better than the other models. And in specific, it performed better on functions associated with viral lifestyle, specifically the tail, head, and packaging, and integration and excision functions. The utility of a learned representational space that can be outputted from a protein language model is that for every single protein, a representation can be produced. And so we wanted to interrogate what these representations captured that might allow the classifier to perform as well as it did. So here you're looking at a UMAP representation, 2-dimensional, of the vector embeddings from the PROT-Trans BFD model that had the best performance. And while it's only in 2 dimensions and the clustering can't be taken for too much value, what you can see is there's a possible partition between the orange, blue, and green, which are tail, connector, and head and packaging, from other colored functions. And that separation is of biologic note because the head, tail, and connector are the constituent elements of a virion structural protein, while the other functions, including DNA and RNA nucleotide metabolism, integration and excision, transcriptional regulation, are associated with the ability to replicate inside of a host. To interrogate the clustering of the families, we developed a distance for category distance by evaluating the family distance for every family in a category against every family in another category. And the single distance metric is the average distance from the families to all other families of the opposite category. And this allows us to create a distance matrix that we then spectrally cluster into 2 groups. And the 2 partitions capitulate what you observe with your eye, which is that the tail, head, and connector categories separate from the other categories. In the spectral clustering, the lysis category also moves with the tail, head, and connector category. And here the families are colored so that you can observe the separation where the tail, head, and connector and lysis are in black and the other functions are in yellow. And when we train a classifier to predict this binary classification, you see performance that is superior to what was observed on any function individually, meaning that the classifier might be leveraging these relationships between these different categories of functions to help make its ultimate classification. And here is 10 random groupings into 5 and 4 or 6 and 3 families, category sets that is used as a comparison to the single partition identified by the clustering procedure. So how can we validate our predictions? We were fortunate that after training our model, frogs had a re-annotation where 46 unknown families received annotation, and our model, our classification model, received an F1 score of 0.76 on this annotation, meaning that unknown families were predicted to be correct based on our classification. And interestingly, 11 families had re-annotation, and 5 out of 11 of those were predicted by our classifier, even having been trained on the incorrect annotation. We used the properties of this classifier to go into a database of viral protein families in the ocean, and there we discovered a novel family of tyrosine integrases in the globally abundant marine cyanobacteria, and I would be happy to discuss this more, and I talk more about it in my poster. Our work provides evidence that protein language models can capture viral protein function beyond sequence homology and serves as a proof of concept for novel biologic discovery. Thank you to Dr. LaBouche Kelly, the Albert Einstein College of Medicine, the Medical Science Training Program, and Dr. Steve Biller, our collaborator on the Integrace story.